All right, so our lab in Miami, we focus on type 2 CMT, specifically type 2E, which, as Steve showed you, is one of the genes that is expressed in the nerve terminal from our nerve cells. So our neurons express this protein called neurofilament, and this is one of the causes of CMT type 2. Here you're seeing a lot of other genes that are associated with different forms of type 2. What I'm going to show you today is focused on neurofilament, but again, it could potentially be applicable, at least the approach, to other forms. Just to understand a little better what we're going to be talking about, for both from my talk and Dr. Conkling's talk, every time we're talking about a gene, we have to always remember that for most genes, we always have two copies, right? We have a copy that comes from our father and another copy that comes from our mother. So if not all, most of our genes have two copies in our genome. When we talk about CMT2s, we are assuming that not only those are genes that are causing problems with the axon of the Schwann cell and of the axon of the neuron and not the Schwann cell, but also that a single change is enough to cause disease. You guys also hear about recessive or type 4 CMT. Those are the forms where you have to have changes on both copies to develop disease. And that's important because that means that for type 2 patients, we always work in a situation where we have one copy of the gene that's not unaffected, so what we call wild type, and one copy with the genetic problem, with the mutation. So one approach that we are exploring with other groups is what happens if we just turn off the bad copy and keep the good copy working. So that's basically, in simple terms, what our approach is, and Dr. Conkling's group with CRISPR approach is. It's basically silencing specifically, or as specifically as possible, the affected copy of a gene causing CMT2. In our case, again, we're focused on 2E, but this approach could potentially be applicable to other types. And the only thing that's really important to determine is whether having a single copy functional is enough to have normal function. Because we know that some genes are very dose sensitive, meaning if you have too little or too much, you run into trouble, and PMP22 is the classical example of that. But we know that NFL, there are many people walking on the streets here in Boston that have a mutation that's turning off one of their alleles or one of their gene copies, and they are fine because they have that healthy copy kind of working. So we know that for neurofilament, we can definitely get away with turning that bad copy off. And there are essentially very likely other genes in the 2 category that will be the same case. All right, so our lab really have started the work using stem cells to study CMT. And when I'm talking about stem cells, I'm not talking about treatment. So we're not injecting stem cells in patients to make those cells into something that will make patients better, because that technology is not there yet. But we use the stem cells to model the disease. And the reason we do that is simply because if we have a patient and want to study a neurological disease, we can take a blood sample and look into their blood cells. We can take a skin biopsy and look into their skin cells, or even a muscle or nerve biopsy, but we can't really take nerve cells from patients.
Recording in progress.
Okay. So that really has been a very hard block to progressing research in CMT, is the fact that we couldn't study patients' nerve cells. But then in the early 2000s, a Japanese scientist figured out a way of getting cells either from the skin or the blood and genetically manipulating them to turn them into stem cells. And that's what's called iPS cell, or induced pluripotent stem cells. And that's what our lab does. We collect samples from patients, either in skin, usually skin, and then make those stem cells. And then with further work, we can make them into nerve cells. And those are actually human CMT motor neurons. And with this, we can then study what's wrong with them, what are the main problems, and then also test treatments before getting to a patient. So this is what our work has been, and this has all been funded by the CMT STAR initiative. We found several different changes in CMT2E human motor neurons. So those neurons, which here's the cell body of a neuron, they get filled with neurofilaments. So neurofilament kind of gets stuck in their cell body. Here is a control nerve terminal or axon, or a bunch of them. And in the patients, we see this accumulation of neurofilament in different points. So we know that neurofilament is not distributing correctly in the axon. And the other thing we found, which is also seen in patients, is if those axons starts to break down because of that genetic lesion, neurofilament starts to leak out of the nerve cell and can be detected either in blood, in patients, or in our culture media of our cells. So we can use this and say, okay, this is another feature of those diseased or affected cells. Now, can we do something that will reverse that? And so we use those features of those cells to test our approaches. Our approach focuses on the use of something called antisense oligonucleotide. It's a long name, but we call ASO for short. And this is based on the fact that since 2016, a ASO approach or treatment called Spinraza or nusinersen has been in use in clinic for our patients with spinal muscular atrophy. And this is a disease where patients, if not treated, would either need respiratory assistance with ventilation, or would actually die from their disease. And we're seeing now those patients growing healthy because of early treatment. And an ASO is basically a small piece of DNA that it's able to link to an RNA, which is the intermediate step between DNA and protein, and block the expression or modulate the expression of that particular region or RNA that's being bound. So this is a way to turn off a potential gene or one of the copies I was talking about. And what we do is we design those ASOs to connect specifically to the bad copy of the gene and turn it off. And by doing that, what we're seeing is here's a comparison. In blue are our affected cell lines, and in purple are control lines, so lines that don't have CMT. And this is the amount of that neurofilament that's leaking in the culture media. And here's another protein called peripherin, which has a kind of a similar use in terms of detecting axonal damage. And we see that once we start treating our cells with different amounts of this ASO, this release of neurofilament or peripherin, so the axonal damage goes down and goes to control levels. So this is an indication that this approach is working, and we can see that we are able to target the specific mutant copy of the gene, and we're hoping to now move to an animal model, which is available for neurofilament, to check what the results of that approach will be. So this is kind of what we do in the lab. And that's kind of for me. And we're going to have now Dr. Bruce Coughlin video talking about his approach, which uses CRISPR instead of the ASO approach. The reason that we selected ASO as our approach is the fact that I think it's closer to the clinic. But hopefully once CRISPR becomes something that we can use safely in nervous system disorders, this will be a great way to approach the same kind of problem. So if we could have Bruce's talk. Thank you.
Hello, I'm Bruce Conklin, and I'll be talking today about CRISPR therapeutics for CMT2, and this will just be a brief update. The take-home message today is that CMT2 is one of the most promising targets for CRISPR therapeutics, but clinical development is unfortunately limited by the rare mutations, sometimes less than 100 patients per edit. Fortunately, we have a new approach that allows 40 times more people to be treated by the CRISPR edit, and this should be enabling for therapeutics. So just given here as an example of one gene that we've been working on, mitofusion 2 or MFN2, which causes CMT2A. As shown here in this cartoon, one disease mutation is what causes the disease and is indicated here in red. And yet there's a wild type gene, which is everybody has, and this one is unaffected. However, when it forms multimers, the proteins actually form multimers. You can see that there are many different combinations which could be contaminated with this red gene. And it's very difficult for CRISPR to target this mutation and at least to repair. It's also this is a very difficult target for pharmaceuticals because of the nature of these complexes. But fortunately, what we found is that selectively deleting genes with CRISPR works very well in neurons, and this potentially could be curative. And that's because when a mutation is made, you can actually get rid of the disease gene, but leave the wild type. And then now the multimers, which would be formed, have nothing to use except perfect forms of the protein, and we have healthy nerves. And how do we know that this is okay? Is in fact because of the superb genetics in CMT allows us to identify people who already have one copy removed through various things in nature, and they were just born that way, and they're neurologically normal. So the experiment essentially has already been done in people removing one copy, but we need to do this experimentally. So just to illustrate the challenge of precise editing in rare disease genes, I'm just showing you here one mutant and one normal gene, which is what people have as two copies. And now if you can target the mutation with CRISPR, you can actually get rid of the mutant. And this actually can work remarkably, even with only a single gene changed or a single mutation. However, this also puts a lot of pressure on that single mutation. And sometimes because of various reasons, that doesn't work out. So be much more, and this is made worse by the fact that this gene in particular, mitofusin 2, has actually over 200 mutations which cause the disease. And so that is a tremendous problem, because if, for instance, you make a one mutation-specific edit, for instance here, that you've only fixed or helped 1 in 200 patients with the disease. So if you imagine there's 15,000 people, which is the estimate for a mitofusin 2 related disease, a mutation-specific cure would only help 75 patients, which is very, very low number for planning a treatment. However, fortunately, there are other ways of targeting one allele versus another. And as indicated here, there are actually these SNPs or polymorphisms that exist in the genome. And because these generally don't cause disease, people actually don't even pay much attention to them. But we found that we can actually actively target them and say, cut out this region in between by targeting these two SNPs with CRISPR, and very effectively inactivate the gene of interest, the disease allele, without the other one. And now the problem is not that we have too few to target, but actually so many different possibilities. And so we've created actually a pipeline to actually identify the types of mutations residing on these common SNPs that would have the most specific, least off target, and so on. And these now, these SNP anchored edits, we have many to choose from, but we can actually choose which ones are the best. And it would seem, you might think it's obvious which ones are the best, but in fact, it involves things that we really don't understand, and we have to test experimentally. So indicated here, for instance, just as an example, is a piece of DNA which is looping around and has three-dimensional structure that we can't see normally. But when we make a cut here and here, these two are close together and then easily cut out the middle, whereas these two, which are actually far apart in 3D space, but actually in linear space, very far apart, that these might have a less of a cutting efficiency. And so we experimentally test now. We're in the process of developing systems to experimentally test the thousands of different edits so we know which one's the best. Now, I'm going to contrast that to a single mutation-specific edit, as indicated here, where now only 75 patients are helped. But now the single CRISPR guide for a pair of guides for a single patient now can be used on 3,000 patients. And so that is a much larger number, and that's indicated by this part here. And then this is additive. One can actually find other edits which capture other variants, and using sort of population genetics, we can actually find 6 CRISPR guides, which should cover about 12,000 patients. And so this is a much better way, potentially, of going about this problem. Now, how common is this in CMT2? We've actually started off our studies with NEFL. Now we have moving into, we're now working hard on mitofusion 2, and we wanted to actually see how this would affect other genes in CMT. And indicated here, the size of the circle is related to the number of patients, with a total of 150,000 people in the US that have the disease. And many of you will recognize your specific gene indicated here. And of course, we're very interested in treating as many of these as possible. So we actually did a literature search and look at experimental data about which of these genes would actually benefit from that. And we were very pleased to see that actually the vast majority of genes which are dominant, causing the types of they have the properties that would benefit from allele-specific editing, and they're all shown here in green. PMP22 is actually shown in a different shade of green because actually it's not. It's a little bit different in the sense that it's, instead of a point mutation, it's got 3 copies of the gene, but allele-specific editing can still be used to remove 1 copy and leaving just the normal 2, and that is something that we're quite interested in. So what could we do for the next steps? We're obviously thinking about the gene-specific programs, delivery, animal models to humanize genes so that we can edit them, regulatory issues. But also we're developed this pipeline, which I described before, which we call allele prospector, which is where we get to test thousands of different edits in a particular gene. And then this actually, by identifying the ideal edit, we can actually then license those or pass those on to groups that want to take it to clinical development. So we're very excited about that, because this pipeline can actually now test many genes, and perhaps that could be useful to many more people. So with that, I'd like to thank you very much for your attention.
All right, so let's move on. I'm going to call Dr. Martin Jeffson from Orthogonal, who is going to give us a kind of an update of their program.
Thank you. Appreciate it. Thank you very much. I'll need that, yeah. My name is Marty Jeffson. I'm the CEO of Orthogonal Neuroscience, and I'm going to give you an update on the development of ORT247, which is we have now in the clinic, and we're targeting, amongst other things, for the treatment of CMT. So I'll go through this slide. Just really the top 2 boxes, as the bottom 2 don't really pertain to CMT, but Orthogonal was formed by a venture investor, Morningside Ventures, by licensing some technology from an investigator in Hong Kong, Professor Nancy Ip, who's at the Hong Kong University of Science and Technology. And one of the things that was licensed in that transaction was ORT247, which is a monoclonal antibody that targets and binds and inhibits the ephrin A4 receptor, which is an axonal guidance molecule, functions very heavily in the developmental stage, but also we believe it's upregulated and functions in disease states as well. And by targeting that and inhibiting that, we hope to increase axonal reinnervation at target synapses, particularly neuromuscular junctions, by 3 distinct mechanisms: protection of vulnerable neuromuscular junctions, axonal regeneration, and sprouting to a degree. We've tested ORT-247 in preclinical models, a lot in vitro and in 2 in vivo models of Charcot-Marie-Tooth disease, and we've seen meaningful improvements in both functional endpoints, electrophysiological endpoints, and also translational biomarkers, which we're quite excited about, and hopefully will guide study in the clinic. We've taken the compound into a healthy volunteer study, a single ascending dose study in phase 1. We've observed excellent safety, tolerability, serum, CSF, and muscle PK thus far, and we've escalated now to the highest dose we're allowed to go to by virtue of the safety studies we've run. That's 1,200 milligrams. And I can say that all of the study is now complete. So we've taken 5 dose cohorts through 85 days post-dose, and we're working up the data at this point. We're planning a phase 2A, or 1B2A proof of mechanism trial in 2024. The bottom 2 boxes just really apply to other applications we're considering for 247 and the fact that we're fundraising to support the clinical work. So here's a little bit more about the mechanism of action. According to the dying back hypothesis, absent intervention, dying motor neurons retract from axon terminals from neuromuscular junctions that innervate muscle fibers. And we think 247 can be active in 3 ways. As I said, neuroprotective for those motor neurons that are destined to die. That's number 1 here. Can help promote axon regeneration for neurons that have already died back. Number 2, and stimulate sprouting from healthy neighbors to recover synapses that have been lost. And that's number 3. So we ran, with the help of Mark Scheidler and the CMTA, 2 in vivo studies at Psychogenics. One was in the C3 mouse demyelinating model, CMT1A, and the NFL mutation model in CMT2E, and that's an axonal degeneration model. And just the scheme, whoops, sorry, go back. The scheme of the study here, we started dosing weekly doses intraperitoneally of 3 different dose levels, 1, 10, and 60, begin at 10 weeks of age in the animals, and continued through terminal takedown for histology. And we did a bunch of interim measurements along the way in life in those models. So here's a very high-level summary of the data. So again, we thought that because of the mechanism of action, 247 would be probably active in the axonal degeneration model, maybe not so much in the demyelinating model, but we were really pleased to see that actually it worked quite well in both models, in both functional endpoints, ePhys endpoints, histological endpoints, and in the serum neurofilament light endpoints as well. And so we have hopes that actually this drug, and to use what Dr. Bouchard said earlier, can be used in combination with gene-targeted therapies to treat patients of all subtypes. So we're pretty excited about that. We are going to start in CMT2 in the clinic in patients because we think that's where we're going to see a signal clinically. That's where it's going to be. But we're hopeful that we'll be able to be utilized in patients of all subtypes. So going very quickly here, I'm just going to give you 2 snippets of the data that we saw on these in vivo experiments. So on the left, in the CMT1A model, it's a measure of forelimb grip strength. You can see that right even at week 10, there's already a profound deficit in forelimb grip strength noticed in the vehicle transgenic animal. And that doesn't get worse necessarily over time. And so we're not really a neuroprotective effect here, but more likely a neurorestorative effect. And you can see that over time, these effects at 12 weeks of dosing become significant. And then here in the 2E model, in the tapered balance beam, we have a significant or a trending effect on foot slips and a significant effect on traverse latency. And then here's serum NFL. And this is in the 1A model. I can say in the 2E model, it works just the same as this. But this picture is a little more dramatic, so I chose it. And you can see no significant trending, but not a significant effect at 6 weeks, but a clearly significant effect at the high dose at 12 weeks. And then at all three doses, or the two higher doses, in fact, at the 20-week dosing point. And so here is what we're doing in phase 1. It's a typical SAD study. Probably the things that are most unusual about it are we actually built a spinal tap for CSF concentrations and a muscle biopsy to measure muscle PK early in the experiment. We've completed all 5 cohorts, and they're out to 85 days post-dose. And this is a very important slide, we think, because so here I'm showing you the PK results from the mouse experiments that I just described at 10 and 60 weeks, 60 mg per dose weekly. Here's the human PK, Cmax, AUC, Cmin, and C average. At the 300 dose, it maps very nicely to the 10 mg, and at the 1200 dose, it maps very nicely to the 60 mg. So we think at these doses, 300 to 1200 mgs in humans, we'll be able to test the hypothesis of whether or not ORT-247 is going to be useful as a treatment for CMT. And then finally, I can also say that we saw good exposure in CSF, which probably isn't as meaningful for this disorder as it might be for other neurological disorders, but we also measured muscle PK, and it's about 1/10 that which we see in serum, which when you think about the potency of the antibody at the target, that should be well more than enough to cover the exposures required for efficacy. So final slide, this is what we're planning to do if we can raise funds to do this experiment, which is a small patient study in CMT populations which are proven genetic mutations and axonopathy subtypes, so CMT2 and others, CMTX, for example. Want to do it as a multiple ascending dose study, 3 doses, 12 to 16 patients per cohort with clinical PK, electrophysiological, and disease biomarker endpoints in the study, and probably a duration of at least 6 months, hopefully out to 12. We can get adequate funding to do this trial. And so with that, I'm going to stop, and thank you for your attention.
So next, we're going to hear from Dr. Smichek from Vanda Pharmaceuticals. She's going to join us by Zoom.
Perfect. I would love to join the meeting in person. My twins got the whole family sick, so I am presenting from home, just to stay safe, to keep everyone safe. My presentation is on the potential ASO treatment for CMT2S caused by IGH-MPP2 cryptic splice variant. And while this is N of 1, a type of study with an ASO treatment, it could potentially be expanded to other IGH-MPP2 patients. And we are working with a patient that had a long journey to diagnosis, identified by Dr. Cassini. A whole exome sequencing led to the first mutation, the missense variant, and ultimately at age 9, which was at age 9, ultimately a couple of years later, at whole genome sequencing revealed a unique cryptic splice site variant. This variant activates a cryptic splice site, causing retention of part of an intron. The resulting transcript undergoes non-associated decay and leads to haploinsufficiency. The variant was carried in a compound head with the missense variant I told you about. And ultimately, if you take a look at the table, the maternal variant, the paternal variant, and the compound head, the patient. While at this point, we are not targeting the missense variant that you see on the left, where there is a loss of hydrogen bonds, we are aiming and trying to rescue expression from the cryptic splice site variant. And on the right, I'm showing you the nonsense decay that is an effect of this variant. So at this point, we compared the patients, we collected skin fibroblast cells, and we compared to a healthy control just to establish RNA levels as well as Western blot, as well as the protein levels. And we do see that deficiency as expected. To remind everyone, although I'm sure you know a lot about IgH-MPP2, it belongs to a helicase family implicated in cellular RNA homeostasis and disease development. I2 deficient motor neurons display morphological aberrations, and I'm going to go into these in a moment, into details. There was a recent work on I2 by Prusty et al. That has shown that I2 shapes mRNA homeostasis and likely acts via cell complex Tregs and ablation of essential cell complex subunit phenocopy these perturbations, which is really interesting as for a group studying I2. So what we do is we illustrate proof of concept experiments with splice modulating ASO tailored to this specific patient's genome and characterize patient-specific CMT2S phenotype through exploratory downstream studies. Now, this is thanks to Dr. Zuckner's group. I am showing here the fact that the missense variant that our patient is a compound head for is present in many other families. We don't know of anyone else with the cryptic splice site variant, but this is the beauty of ASOs. While potent, they target, they are mutation specific. So our ASO, I am showing you the diagram. On the left, you see the loss of I2 protein, whereas on the right, you see the upregulation thanks to restoring activity masking. Basically, it's a steric blocking ASO that masks the site and prevents the NMD and ultimately rescues the expression. So at this point, we designed multiple ASOs. We chose the one. It was a trade-off between efficacy and safety. As some of you know, ASOs with quadruple Gs, for instance, can be more toxic. So it was a experimentation stage where we chose the most potent ASO that would be the safest. We did RNA experiments where we saw around 9-fold relative to untreated control samples increased boost in expression levels, as well as a dose response. I'm showing you a dose response on several different treatments. Our ASO is the same PS Mori technology as the one used by nusinersen. And doses are also within that range. Here I'm testing an actual broader range of doses. And as you can see, 5 has the highest levels, although with already one micromolar, we are pretty satisfied. And to give you, so basically this was the molecular genetics aspect of the presentation. What we are also doing is we are testing and trying to translate the effects of this mutation using human electric neuromuscular junction system. And this is really to be able to phenotype this, translate the effects of these mutations, and ultimately be able to compare the effects and of course demonstrate efficacy with the ASO. So what we have done here is to further characterize the neuromuscular phenotype, we analyze morphology and physiology. I'm just going to briefly highlight the most important aspects. We developed CMT2S patient-specific NMJ model. We generated iPSC stocks for the motor neurons and the skeletal muscle. And what we've been seeing initially is that those derived motor neurons demonstrated an expedited maturation in comparison to wild type, as determined by axonal progression from motor neuron cell bodies, penetration through NMJ tunnels, and second skeletal muscle innervation. Again, we are comparing to healthy controls as well as we have other phenotypes that we can compare against. But this is this specific patient cell, derived motor neuron. They have slightly higher axonal branching. The difference between Charcot-Marie-Tooth type 2S is gradually increasing across time, as you can see on the panel on the right. And we also have preliminary NMJ functional data. We were looking at the number per chamber, NMJ fidelity, and NMJ fatigue index. And what we see is compared to the wild type, the fatigue index representative traits demonstrated quick fatigue, and quantification of the fatigue index showed complete or partial tetanus, revealed an increased fatigue index using the CMT2S NMJ across all test phase. And again, please take a look at that panel on the right to depict that, with Charcot-Marie-Tooth being the bottom. So to conclude, on the molecular genetics, I think that's straightforward. We are targeting that specific mutation with the PS-MOI technology that is now approved by the FDA, and we are restoring activities, so providing rescue on the RNA level and on the protein level. We are also running toxicology studies. So far, the drug has favorable safety profile. We have the safety studies are ongoing, as well as the PK analyses. And we have clinical protocol under development. And on the motor neuron characterization, this is interesting because we could be potentially showing a rescue of different ASO, restoration of the desired protein levels with this model, and also we could compare different IQ mutations and the consequences using this model. So hopefully, if this could be shared across different groups potentially. And of course, we will continue analyzing this and other cells from different patients with IgH and DP2 mutations in order to rescue the protein levels. And with that, I would like to thank you, and hopefully I will be able to update you with more from the clinic in a couple of months.
And I'm going to move next to Dr. David Goldstein from Actio Bioscience, who's going to give us also a update on their program. And with that, with the last talk, and hopefully have some time for questions.
Well, thank you. It's really a privilege to be here and to share a little bit about the work that we're doing. Before I go into details about the program that we're working on, just a little bit of context about the company and how I came to work at the company. So first about myself, I'm a human geneticist by training and until recently by practice, which means I did not spend my career as a drug developer. My work in human genetics really focused on trying to discover the causes of genetic diseases, and my group discovered a number of them, and also focused on trying to diagnose genetic causes of diseases in individual patients. And as part of that work, I spent a lot of time communicating genetic diagnoses to patient families. And what I found is that in the vast majority of cases when I communicated a genetic diagnosis to families, what I told them is that we knew the cause of disease, that told us something about prognosis, but unfortunately there are no effective treatments. And that really is what I normally communicated. I, of course, found that increasingly frustrating, as of course even more so the families did. But what I found in a way even more frustrating is that a number of the underlying causes of disease looked to me like tractable propositions for drug development efforts. Like you really could develop an effective treatment if you brought contemporary drug discovery efforts to bear on those genetic conditions. But many of them were actually not being worked on because of a perception that it would be too expensive to work on them given the number of patients. I felt from the academic perspective that orientation was mistaken. And I actually spent some time to try to convince companies that I was right and they were wrong. And they said, well, you're a great human geneticist, but you're not a drug developer. So eventually I said, well, you know what? I'm actually going to stop being a human geneticist and I'm going to be a drug developer. So I started the company really to try to focus on those opportunities where it looked like, even if there are not that many patients out there, you'd have a very good chance of developing a truly transformational treatment. That is the focus of Actio Biosciences. The way that we work is outlined here. We identify the cause of disease as a clearly explainable underlying cause where the mutations effect are well understood. We then characterize ourselves those effects of the mutations to make sure we understand the way they cause disease and that we have some way of actually testing potential treatments. And then we move on to actually assess how potential treatments work in appropriate so-called preclinical models, and eventually move on to trials. Part of the way I convinced funders to go after these rare diseases is the belief that in some cases the treatments we develop will actually have relevance beyond just the rare disease. I'm not going to talk about this here, but this is something I think often happens and needs more attention. So just a little bit about the first lead program in the company. It's focused, of course, on TRPV4, which is a cause of a rare form of CMT. CMT2C, although quite rare. There are a number of patients even here that have CMT because of mutations in this channel, and it fits our paradigm because the way the mutations cause disease is understood, and it actually should be addressable with traditional drug development efforts, that is small molecule modulation. So what happens in this channel is mutations activate the channel. The channel is a channel that allows calcium to come into cells, and we know for sure that the presentation actually results because activating mutations allow too much calcium to come into cells, and that causes all sorts of problems. We actually understand a little bit about exactly what cells are important in causing the problems. In addition to a form of CMT that activating mutations cause, there's another set of diseases that affect the skeleton that I won't be talking about here. The way we try to work in the company is really comprehensive so that we will be able to develop a treatment that applies to all patients that have CMT because of mutations in the target that we're working on. And so what that means is that we start with every single known disease-causing mutation. We characterize the effects of all those mutations in the lab. We've been able to show it sometimes actually takes some work to do this. You have to actually move between different types of cells, but we've been able to show the activating effect of all the mutations. Sometimes they are big activators and sometimes they're modest activators, but we've shown activating effects of all of them. And then for the compound that we've developed in the company as a potential treatment, we've actually assessed how well that compound works against every single one of the activating mutations. We've been able to show that our compound is actually able to inhibit the increased activity of all of the known disease-causing mutations. And what the meaning of that is that in principle, the treatment should be usable for every patient, at least as far as we can tell, based on work that we can do in the laboratory. So then the next question in the drug development process, you show that you've got some activity in simple systems where you've got the disease-causing target expressed in a cell. It does something. You can counter that with a compound. The next thing you have to do is ask how well the treatment works in a so-called preclinical model, where you build, for example, a mouse model of the disease. Of course, many people here know people with CMT, so you'll recognize there are some differences between those people and the model that we're going to describe. That means that sometimes what we see doesn't translate. But the way we work on models is we make sure that the underlying cause is the same. So it's a mutation in the mouse that actually causes the phenotype we're looking at or the presentation in the mouse that does the same thing in humans. And then we look to see whether the presentation you get in the mouse is similar. Both of those things happen here. And that really gives you about as good a chance as you can have that what you see in the preclinical model is going to translate clinically. This is work that's been done by our really phenomenal collaborators at Johns Hopkins, Charlotte Sumner and Jeremy in her lab. And they have characterized multiple models for mutations that cause CMT. And I'm going to describe what happens with one of these and what happens when you treat this mouse with an inhibitor of the channel, maybe. There we are. So what you can see here is a mouse who is affected by the mutation. The mouse does not move around as much as a mouse typically does. The head is drooping. The forelegs are very weak. So this is an affected mouse with some similarities to the presentation in people. Treated once with an inhibitor of the channel. And as far as we can actually tell, that mouse makes a complete recovery, as far as we can tell. This is a truly striking result. I actually was inclined to believe this result right away because Charlotte Sumner is really one of the best research neurologists around. But we have also reassessed this in an independent lab. We moved we characterized this model together with investigators at the Jackson Labs. We've actually seen that the model looks the same in the Jackson laboratories in Maine as it does in Charlotte's lab, and we also see the response to treatment. This is a really exciting indicator that we should actually be able to see a beneficial effect in people, and also the possibility that it'll be a relatively rapid effect. Now, Charlotte actually has done a tremendous amount of work to understand the biology of why we should get such a quick response to treatment in the mice. And what we think is actually happening is that this is not an effect of the mutations in neurons. a lot of work actually suggests that what's happening is that this is an effect of the mutation in cells that are in the vascular system that actually protect the nervous system from things in the vascular system that should not get out of the vascular system. So the mutation affects this barrier between the vascular system and the nervous system. Some things leak across that barrier, and we think that's what is actually interfering with neuronal function. You introduce the inhibitor, that barrier firms up, and the mice get better. And this is what we actually are hoping will also happen when the treatment is introduced in people. In order to be able to test a treatment, you have to understand the disease. This is one of the real challenges, as I think everyone here will know, of rare diseases, is that they're often not well characterized at all. This was another one of my major frustrations in academia. A lot of them are too rare, and people haven't really done the detailed studies to understand exactly what the disease looks like. And if you don't know what it looks like, then you actually don't know how to run the trial to test a treatment. So we've been able to pull together a terrific set of collaborators to really help us try to characterize the so-called natural history of this particular form of CMT, CMT2C, and a natural history study has been planned out primarily by Brett McRae and Charlotte Sumner, and it will be initiating at Johns Hopkins probably in January. One point, yeah, and I would like to acknowledge there's been very strong community support, encouraging people to participate in this and encouraging investigators to start the study. I would also like to point out that one of the things the Hopkins group will be doing is offering to characterize anyone's mutation. So as you probably heard earlier, sometimes you get back, when you have a genetic report, a clear answer. Sometimes you get back nothing, and sometimes you get back a maybe. Those maybes, when they're in TRPV4, we actually have a good way to assess whether they really are responsible by characterizing the functional effects of the mutations. So anyone who goes through the Hopkins study who has a maybe, they actually can ask the Hopkins group to characterize the mutation, and then they'll work with us, characterize the mutation. It may not be a definitive answer still, but it might add more information to that maybe. So this is actually ongoing, and I would encourage anyone who's interested to get in touch with the wonderful folks at Hopkins. I do want to really strongly emphasize that the way that you introduce a new medicine is by working as a team. No individual, no lab, no company actually can do this alone. And we have been really privileged in having just the best team possible inside the company and outside the company. And as part of that, I would like to really give my heartfelt thanks to the CMTA. When they heard about what we were working on, they jumped in right away and asked to help in absolutely any way they could. We ran a focus group to meet some of the individuals with CMT2C to start that long process to understand the condition as well as we possibly can, so that we could actually know how to evaluate our treatments when they are ready to be evaluated. And that is really what I wanted to share. I think there's going to be a Q&A session after. I'm happy to address any questions as long as you don't ask really hard questions. But I really would just really like to emphasize that we really do see this as a community effort. We are here to try to help. We want to be as responsive as we can. If there's anything that you think we need to be doing that we're not doing, please let us know absolutely anytime. Thanks so much for your attention.
Thank you very much, Dr. Goldstein. So I know we're supposed to break now, but I'm more than willing to be here and answer questions, and I guess Dr. Goldstein as well. So whoever has any questions? Yeah.
I'm not a scientist. I don't pretend to be. I'm not a scientist and don't pretend to be, and I may have missed something in the earlier presentations, but in all the conversations about gene editing, is that designed to stop the disease from progressing as opposed to helping, once the disease has progressed to a certain point, is there then hope that the nerves will regenerate?
Right. So this is not only for gene editing, but I think any approach for CMT, be small molecule, gene editing, gene replacement therapy, it really depends on when you're treating the patient, right? So there are certain things in the nervous system that, at least with our current technology and medical knowledge are irreversible. So if you have axonal loss, so loss of your nerve fibers, and that creates scar tissue on that nerve, the muscle changes in morphology or in composition as well. So those are things that will be very hard to be reversed. So if you are only able to treat a patient that already has those changes very well established, it's very unlikely that we'll be able to reverse things. Having said that, we know that our peripheral nervous system has way more plasticity than our brain or our central nervous system. So it's possible that we'll be surprised by how much actual reversal or improvement we can see. But just from a realistic standpoint, and also based on experience from other diseases, like I mentioned, SMA or spinal muscular atrophy, and we see that, of course, it's way more effective to treat the babies that are born with the disease than treating the adults. But even the adults, we do see gains happen, but they're way less marked than in younger patients. So it depends on timing. There's a potential for improvement, but at the least, our expectation is to stabilize the disease.
I had a question just across the board about everything you've shared with us. I think a lot of them are right now targeting people with dominant mutations. And can you just say a little bit about how these apply to or might someday apply to recessive?
Sure. So the recessive session was actually next door, type 4. But just so to kind
I have a 2E recessive. So.
Oh, okay, so you're very special. Because, yeah, so 2E, it's mostly autosomal dominant, but there are a few cases where recessive forms can happen, but very small margin. So for the recessive forms, we believe that gene replacement therapy is kind of the obvious way to resolve it. And again, spinal muscular atrophy showed that this is possible. It really, with the current way that we have to deliver genes to our body, we're limited by the size of the gene. So if it's too large of a gene, it's harder to do it. And the other problem is what Dr. Coughlin alluded to is it's very hard to think of a sustainable platform where we could do this for every single recessive disease. So things will take time from one success story to another success story. But hopefully, once we get better ways to deliver genes to the body, I think this will then really explode in terms of how we can treat recessive diseases, or what we call loss of function diseases. It's basically replacing what's missing. Yeah.
And Dr. Goldstein, how does your thesis about the barrier between vascular and nervous apply to this? Because it seems like structurally that might be a little more.
Sure.
For those that want to take a break, there's snacks out there. We're going to start the next session at 3:45. Maybe a conversation one-on-one.
It looks like this mechanism might be quite specific to CMT2C. It's the only channel, and it's the only example with definitive evidence that it is leak across that barrier. And of course, we're very interested in exploring if there might be wider application. But right now, we know that's what's happening in 2C. We don't know for sure it's happening elsewhere.